To start us off, Kim Boekelheide from Brown University is going to provide an overview of the process of transformation in toxicity testing. Kim?
Well, this is like old home week. I'm glad to be back to emerging sciences and be able to present to you. I'm really quite honored to be here. I am not a risk assessor, and I think I've been chosen to kick this off. Let me say, I hope I've been chosen to kick this off because I am not a risk assessor. So that allows me, hopefully not to get all the way out of the box of thinking in risk assessment, but at least to expand the boundaries of what we're thinking about as we try to apply these new tools to the risk assessment process. So I've participated in, various risk assessment actions. I know what the boundaries are to some extent, but I'm not in the field, so I'm not constrained by it. So I hope by the end of the talk you don't think it was a mistake to choose someone who is not a risk assessor to make this presentation. My charge from the committee was really threefold. The small thing, give a visionary talk, get everybody excited. Be inspiring. I'm supposed to define systems biology for everyone. And then finally, I'm charged with going beyond just a nice science review to give guidance on breaking some of the frameworks that we have been stuck in historically. So I'm taking this charge seriously, and again, I hope it's not too far outside of the box. I'm going to start off with a very specific example because I believe we are in a time of transformation in biology, and thus the title of my talk. So this is a time of transformation where the new biology, our understanding of biology as a quantitative science at the molecular level, is replacing the phenomenologic biology of the past. And transformation times are also times of disruption and discontent. And I think there is a lot of discontent and disruption in our fields right now. I'm using this specific example of bisphenol A. You've all heard it in the news. You probably know about this. But there are significant controversies in bisphenol A, and I'm going to label a couple camps of the controversy just to illustrate what is happening with our science. And so there's an endocrinological point of view and a toxicological point of view, and these are really quite different. So bisphenol A is an estrogen receptor alpha binder, but at a very low affinity. So its biologic action at least low levels cannot be through this particular mechanism. However, in vitro systems, it has effects in the nanomolar range. In non-GLP animal studies, there are effects at the nanogram per kilogram level. In GLP animal studies, the effects are seen in the milligram per kilogram level. All of these are true. These facts are, in fact, facts. They are seen and known. They're in the literature. In fact, there's a gigantic literature on this compound. But there is real disagreement as to what the meaning of these facts is. And in some sense, that controversy is driven ultimately by a controversy over the concentration of bisphenol A at target tissues in vivo. So what is the difference in view about bisphenol A? There's low-level effects from the endocrinology point of view in the nanomolar range. The toxicologists see effects up in the micromolar, millimolar range, and they integrate those effects into a response pattern emphasizing the high-dose range. Is there molecular adversity from an endocrinological point of view happening in the nanomolar range, and a physiologic adversity happening at the higher dose level? And, this raises some significant questions about what adversity is, and we'll return to this point later on. And I want to put this controversy actually into the context of background exposures and biology in a larger sense. So this is a figure from a relatively recent paper looking at the percent of abnormal oocytes in meiosis II in a mouse model, comparing the response to bisphenol A in animals who have been exposed to a phytoestrogen-rich diet, that is a diet soy, the black bars here, or a diet that is casein and therefore phytoestrogen poor. And if you look at the 0 microgram per kilogram, that is no bisphenol exposure bars in this figure, there's a real difference in the percent abnormal oocytes at 0 based on the exposure to phytoestrogen or little phytoestrogen. And all of the response that's seen with bisphenol A is actually within the range of experience of these animals who are either phytoestrogen exposed or not phytoestrogen exposed. Therefore, the linear dose response to bisphenol A shown by casein background animals or the U-shaped dose response shown by phytoestrogen exposed animals is all within a range of experience based on what the feed was of these animals. And these are significant effects upon a background which itself produces very significant differences in this animal model. Again, we're going to come back to this question, but it raises issues around what is adversity, how do we interpret effect, and what is normal. So we go back to a very basic graph in toxicology. We all, I think, will agree, I hope we can agree on this kind of behavior. As you increase dose, there's an adaptive response. At a certain threshold, a toxicity threshold, that adaptive response is overwhelmed, and we have a toxic response. Does anybody disagree with this statement? Good. I think we can all agree on that. Okay, that's fine. We can all agree on that except what is normal. We already saw in the animal study that phytoestrogen or no phytoestrogen produces very different normal. And what is adverse? How do we define adverse? Where do we draw that line or identify that point of toxicity threshold? And it's going to be different depending on what our worldview is, what our experience is in the science that we do. And then there's all the things we actually don't understand or know about, like epigenetic effects, and I'll return to this point also later in my talk. The unknown biology that we know is important, that is influential, that has effects that we don't know yet how to incorporate into our models and our understanding. So my point of bringing this very specific example forward is really to emphasize the fact that we are, in my mind, in a time of transformation. There's a lot of discord, disagreement, and contention. And I think in the toxicity testing field that we're all immersed in, we've actually already done our transformation. The accepted paradigm that we had for the last 50 years that animal testing is going to drive our risk assessment process, suddenly doesn't seem right anymore. We're using it still because we have to, in a sense, because we know how to do it, but we're all searching for that replacement, and in fact, we're moving very quickly to trying to find a replacement that will do a better job for us in doing risk assessment and being predictive. So the truth that was animal testing, that we believed in, that everything worked in a coherent system in, is not true anymore as science, and we're looking for a different truth that we can hang our hat on. And surprisingly, I think that transformation has happened quickly. It's not been agonizingly slow, as some transformations can be. And I think we're in a new world. We're just needing to find what the parameters are in that world. All right, so what I want to do actually now is contrast the previous truth of a testing risk assessment system based on animal models, as we know it from the Red Book, with what an ideal toxicity testing system would be. So in this slide, the black box is our ideal test system. I'm going to try to describe some of the parameters in this test system that make it a better ideal system that we want to have as part of our system as opposed to the traditional animal-based toxicity testing and risk assessment approaches. And the whole goal of this black box approach is to identify whether an environmental chemical is safe or not safe. So here's our traditional risk assessment approach: hazard identification, dose response assessment, exposure assessment, and risk assessment. And in my view, the problems with this approach is that it basically ignores our current understanding of modern biology. It does not incorporate the complexity of our emerging molecular understanding of the system of biology as it works in cells and in our bodies. So it ignores biology. It's very linear. You start at the beginning, you sort of go through the process, and you get to the end. It's very simplistic in terms of its architecture and structure. And the whole goal of this process traditionally has been to get just one number. You go through all of this data generation accumulation, and all you're really looking for is the no-L and the low-L. After doing all that work, and you don't incorporate the richness of understanding that you've generated by the work that you've done. So that doesn't work. So what are we looking for? We're looking for a biology-based testing system that is interactive, that is complex, and probabilistic. So the now, what we're doing is driving over animals at very high dose and leaving them on the road. And the future is a systems biology, complex biology approach that takes into account everything that we can know about how these complex cellular machines work. The past is too linear. Start at the beginning, go to the end to get your one number. I love this figure from NIEHS. It's the strategic planning figure. Forget the words. It's just so colorful. Everything moves in it. That's what we want our test system to do. It needs to be active, interactive, moving, to be informative and pliable and responsive, and have lots of moving parts. What about the simplicity? You know, simplicity is nice, but unfortunately biology is complex. And, you know, there's a really, really nice TED Talk by Eric Berlow I encourage you to go and look at. I put the website here. I previously actually tried to show this. It's a 3-minute TED Talk. It's a fantastic TED Talk, and I failed because of electronic, screw-ups in the presentation, so I didn't do it this time. But, biology is complex. We need to incorporate that complexity. But in fact, after you have all that complexity in hand, what will emerge from that complexity, I imagine, and I think is true, are the important nodes of response that are actually key, that are characteristic of the response of the whole system, so you can study those and be informed about the response of the whole system by looking at a few things, or a relatively few things, after you understand how it all interacts. So in fact, the system we need to go to has to change from being very simplistic to being complex, but it will also then become simplistic again as we understand, but it's the simplicity of knowledge, not the simplicity of ignorance. And then finally, rather than looking for one number, we actually have to incorporate everything that we learn into our estimates of safe and not safe, and into our uncertainty about the safe and not safe estimate. And so I quote here from a book, Probabilistic Reasoning in Intelligent Systems, Networks of Plausible Inference. I have absolutely no idea what that means, but that's what we have to do. And I'm not a quantitative, mathematically oriented person, but we have to be probabilistic in what we're doing. We have to incorporate the uncertainty of our investigation into the final determination of safe and not safe. So our goal in our ideal test system is to have this very colorful, moving, active, interactive system that's biology-based, interactive, complex. Initially, we need to generate that complexity in order to find the simplicity of understanding, and probabilistic. And our goal is to find out what is plausibly safe and plausibly not safe in terms of a chemical exposure. All right, so that's my visionary introduction. Systems biology definition, and this is really critical, I think, for this meeting. It's an interconnected network of events, systems biology is, that is predictive of the emergent properties of the system. So here we're looking at a time scale, seconds, minutes, hours, days, and responses. And there's going to be a time scale to the integration of the response with an exposure, and it's a systems biology integration network response. So we have a ligand receptor activation, kinase activation, nuclear receptor translocation, transcriptional activation, and metabolic activation. There's a whole sequence of events that takes place in the cell. These are interactive, interconnected, dependent upon each other, and in fact, all of those interactions are predictive of a physiologic endpoint when the knowledge system that we have is sufficiently informed so that we can understand the basis of those interactions. So systems biology, for our purposes, is an interconnected network of events that are predictive of emergent properties tied together. And this is actually important, as you'll see later on in my talk. You've all seen this figure before. This is the National Academy of Sciences 2007 report figure encapsulating the proposal about the toxicity testing that we need to do. And there are really four major components here: chemical characterization, toxicity testing, dose response, and exposure context and risk context. The committee in 2007, and I was a member of this committee, was aware of some real problems and challenges that were needed to be faced in order to take that vision encapsulated by that last figure and translate that vision into a working toxicity testing system. Some of those challenges are around assay design and development. What are the platforms? What are the assays that we're going to use? How do we identify metabolites in vitro systems? How do we develop the tools to interpret the systems biology information that is massive that we're generating and the connections between the different kinds of data streams that are being generated? And then Isla referred to this point: how do we actually talk to each other across the boundaries that we've all been trained in order to have a coherent conversation about all of the components that actually encapsulate this toxicity testing system? So there's an educational component. So lots happened in the 5 years since the 2007 report came out, and I want to speak to some of that progress that has moved the visionary figure from a visionary figure to an implementation plan. And so this is, again, you can see across the top there the four components that are in the figure: chemical characterization, toxicity testing, dose response, and exposure standard risk characterization. But this approach has now been elaborated on, and one of the proposals that's come out is that a prototype, using prototype models to exercise this system, and in other words, choosing compounds, cell types, targets that activate the systems biology network so that we can start to exercise and work the system that we're developing. That's an approach that is happening and needs to take place, and it goes across all of these. The components will be developed as the information is rendered from doing the tests that we can do, and incorporate human genetic variability, human susceptibility, adversity as a question, mixtures, and the things we don't really know about, like epigenetics. And all of this needs to then be quantitated. We need computational systems biology pathway analysis tools, and then how do we extrapolate from the in vitro exposure concentration response to an in vivo response. And then we need to do this all in the context of what the human exposure is and monitoring humans and correlating with human response. Throughout this, we have to validate our test systems and our approaches and educate ourselves so that we can talk to each other. So this is a very complicated process of implementation. This is going to take us 1 or 2 decades. But it's a really wonderful process of discovery for science, for us, and it's broadly applicable. Here are some examples of some case study approaches. Some of these are already being pursued, so PPAR alpha, P53, estrogen receptor. There are groups that are now working on these targets and compounds in order to start to exercise all of the components of the new toxicity testing system. Here's an example of P53. This is all going to be put into a quantitative framework so that we can predict from the responses where things start to go awry and ultimately identify the point where an adaptive response is replaced by an adverse response. This is just one example of how the system ultimately is not going to be linear, but is going to be repetitive and interactive. This isn't quite as colorful as the pinwheel, but the process here is going to be cycled through multiple times with different approaches and different tests so that the accumulated knowledge is used to inform the point of departure in the in vitro system so as then to extrapolate to allowable or safe or plausibly safe in vivo exposure levels. So this is a complicated machine that we're all engaged in developing, and it is going to take us a number of years. I want to go now to the third part of my charge, which is to use my very big box of lack of knowledge about risk assessment to make some comments about what we really need to be thinking about and maybe breaking some bonds and maybe saying some stupid things, but that's okay. And I'm going to make comments in 4 different areas. One is around screening, ranking of chemicals, and the point of this is to determine which of the chemicals are more or less likely to be hazardous. Hazard identification, that is to determine whether a chemical causes a particular type of effect. Mode of action analysis, that is, looking at how a hypothesis of how a chemical does its activity. And then finally, the question of adversity. So screening, ranking of chemicals in terms of prioritization for how we are going, which ones we're going to test and how we're going to test them, we already do that. We do that in a very disorganized way. But we already do it based on public concern, based on politics, economics, QSAR, exposure, ToxCast, Ames test, animal test, production volume. There are lots of triggers for the ranking that we currently do for the chemicals, the 100,000 chemicals that are out there. And the big question is, do we actually go from this system, which is chaotic in some sense, driven by lots of different influences in terms of ranking, to a screening, ranking, prioritization protocol that is identified and defined? And is this identified and defined screening, ranking, prioritization protocol similar or different from the real test system that we're using? And I guess, finally the question is, and I think for a while people thought this, is our screen, in fact, in vitro and our real test in animal? And I hope we're beyond that. I'm confident I'm beyond that. I hope the rest of the world is too. So to put it simply, there are really 2 models for this. If we actually go to identify a screening, ranking, prioritization protocol, is that screening, ranking, prioritization protocol a distinctly different basis for assessing the chemicals? In other words, is the conceptual and technical approach for that screening process different than the real test? Or is the test system have a subset of tests that are under the same conceptual framework, the same technical approach, that are the screening elements in a holistic test system. I actually think this is a really important point. And, why does that matter? So if we go back to our systems biology definition, everything's interconnected in systems biology. We have our responses for ligand receptor activation, kinase activation. These are tied into the later effects on the cell and ultimately to the phenotypic readout of that cell. Systems biology gives us the interconnection and technical approach, if we can capture all of this information together, to put the screening system under the same umbrella, within the same framework as the test system itself. So I would very strongly advocate for a screening approach that is within the conceptual and technical framework of our ultimate test system. Why? Because this is parsimonious. It allows the test system to continue development, and we can spin off different screening elements because they're justifiably better over time. It's more likely, if you use the same conceptual and technical framework for the screen and the real test, that you'll end up with the same rank order of the compounds in the two approaches. And it's a whole lot easier to defend one system than two. And in fact, if you have two different systems, if you're setting up a model where the screening system is different from the real test system, what will happen is they'll be played off against each other rather than working together to support each other. So I think this is really important in framing the approach to developing a screening protocol, if that's what happens, that be very clearly articulated as emerging out of the context and, in fact, out of the same tests that are used for the test system. So that's point number one. What about hazard identification? And hazard identification is whether a chemical causes a particular type of effect. And what is meant by a particular type of effect? In an in vitro test system, we need to ask ourselves, is this particular type of effect that we are imagining, that we're predicting, an in vivo endpoint or not? Or are we just trying to do an assessment that allows us to be plausibly safe or plausibly not safe with our test system? And I would argue that we should not try to use our test system, or we don't have to use our test system to actually extrapolate to in vivo endpoints. And I'd like to explain that. So on the left of this slide is, and it may be a little hard for you to see, is a paper from Bas Blaubauer, and he's been working in this field for decades. And he's proposing a tiered approach to in vitro testing. And you'll see in his tier that he actually has organ, tissue level, in vivo level types of tests. So in vitro livers or in vitro muscles. And in fact, in the National Academy of Sciences 2007 report, in the toxicity testing module, there was the targeted testing component because the committee ultimately, as a group, felt that there were unknowns that we could not tackle clearly with in vitro approaches alone. So we needed something else there, which was called targeted testing, which included maybe some animal models, maybe some lower animal model complex systems, but stuff we didn't understand that was just too much to handle maybe in an in vitro system. I'd like to argue for a different approach and a different way to think about this. What we want to capture in our test system is the full range of biologic response that is possible in a living organism. So we use in our brains different pathways to do the signaling, to do the processing than we use in our livers. But if we capture in our test system all of those pathways and the full range of response that is occurring in the in vivo setting, then we're covering the biologic landscape, response landscape. And if we are covering that biologic response landscape and testing chemicals in that system that covers the biologic response landscape, and then doing a risk assessment against those responses, then we are plausibly safe and not safe. And we can develop this knowledge about the full range of the biologic response landscape in really two ways. We can take an agnostic approach, where we just start testing, we develop models in vitro, and we just accumulate the knowledge of all the pathways and all the responses. And over time, ultimately, if you do this for an infinity amount of time, you will have covered all of the biologic response landscape. All you need is tremendous amounts of money and tremendous amounts of time. And there's value in taking this kind of agnostic approach to knowledge generation. And the value is you don't have a preconceived notion of what the boundaries of response are. And therefore, you can expand that biologic envelope to its ultimate extent, given enough time and enough money. You can also take a very directed approach to generating this knowledge and use the expertise of individuals who are focused in particular areas, let's say development, reproductive toxicity, neuro, immuno, and allow those individuals with their expert knowledge to design systems in vitro that are going to give as much of the response pattern landscape that they're familiar with in their area of expertise. And this might get you to the boundaries of the biologic response landscape in their area a little faster. But you won't get to the biologic response landscape that you don't know about if you do that. So maybe the best approach is a parallel approach. You have some people working on system generation without prejudice, agnostically, and you have some experts working to develop their response landscape. You put them together, and over time, you're increasingly expanding your understanding of what that biologic response landscape is. So the goal of all of this understanding of the biologic response landscape is to understand the systems biology integration to physiologic endpoints. And for that risk assessment in vitro, the physiologic endpoints of concern are cellular perturbations. That's the integration that we're doing if we're taking an in vitro toxicity testing approach. We're integrating at the level of the cell. We should regulate on those integrated systems biology inputs into the cellular physiologic response. We don't have to extrapolate from those cellular integrated responses to in vivo. We can, we don't have to for the risk assessment purposes. And in fact, I would argue that we can get bogged down in that step of extrapolating from cellular physiologic responses to the in vivo organ, tissue, whole organism response. It's unnecessary to do it, and we could spend a lot of time spinning our wheels about how that extrapolation happens, even though all our data is being generated at the level of the cellular physiologic response. If we are covering in our test system all of the biologic response landscape that's available in vivo, if we're covering it in vitro, we don't have to do that extrapolation. It's nice, it's informative, it will be useful over time, but we don't have to. So I would argue that we should avoid that unnecessary complication. And in the EPA rubric, what this means is that forget the tissue organism tox endpoint for the purposes of risk assessment. Let's focus on what we're generating as data, which is systems biology integrated physiologic responses at the level of the cell. And in fact, this is where science is going, and medicine is going. If you look at any of the cancer kinds of treatments that are currently being pursued, they're all at the level of cellular response pathways. That's where all of the action is. And over the next decade or two, if you listen to Leroy Hood, that's all we're going to be doing. We're going to be understanding responses at the level of the molecular interactions within cells in the body. And for cancer, it no longer matters what the type, where the tissue or the type is that cancer arose in. All that matters is its molecular phenotype, and that's what we're actually treating against. And so over time, there's going to be progression of epidemiology and medicine to a molecular epidemiology and molecular medicine, and toxicology at a systems level and risk assessment at a systems level, to a coherent understanding of systems biology and toxicology, of health and disease, and that is what we will be doing ultimately our risk assessment on. So I have only a very brief comment on the mode of action analysis question, because there's a lot of talk around this, and I think there's a lot of misunderstanding, including on my part. So I tried to understand it. So what is the mechanism of action? How does it differ from the mode of action, toxicity pathways, and adverse outcome pathways? I find this very confusing. So what I would argue we should do is focus on toxicity pathways, systems biology understanding of cellular responses, and their integration. Enough said. I don't even really understand what adverse outcome pathway means, although I've heard it talked about. All right, so let me sort of turn to my final point, which I think is actually a particularly difficult one, around adversity. And I call this the third rail of the new toxicity testing. And I really think it's a topic, there's been some significant discussion about adversity. There's been an Elsie Hesse committee that's focused on adversity. I've written a paper about adversity. Others have as well. But it's really a tough topic. And my sense is actually we can't grapple with adversity until we start to have some of this systems biology level of knowledge that we need to be interactive and interpreting in order to actually identify what the responses are that make cellular responses adverse. So in a sense, we're trying to label adversity responses in a vacuum at this point. I think it's important conceptually to think about, very important, but ultimately it's going to come down to a data-driven process, and we don't have the data yet. So adversity is really a lightning rod. For industry, the fear is that any little kind of blip in a cell is going to make chemical X be plausibly not safe, and therefore they can't make anything and they can't sell anything. Back to our endocrinology, toxicology conundrum, the endocrinologists fear that those little molecular blips at low dose they think they're important, and if we ignore them, that in fact we're missing important biology. And the toxicologist's fear is that if we pay too much attention to those very low dose phenomena, in fact we miss what organisms can do and what cells can do, which is adapt to exposures. And so we're paying attention to the trees as opposed to the forest of the global response. And we all fear that what we don't know is going to kill us. So I want to take a little bit of a sidestep here and talk about epigenetics. I've mentioned it several times as something we don't really understand, and it's true. I think we're just beginning to understand epigenetics and how it influences us, and how we're going to incorporate those phenomena into toxicity testing, I think is really an unknown. And so I have the example here of the Dutch hunger winter. So this was famine conditions in the Netherlands at the end of World War II. And there's been really just wonderful studies by Barker and others, his group in the United Kingdom, that have looked at the consequences of that famine condition for those who were in utero during that famine in terms of their disease outcomes later in life. And in fact, they're very significant. So we're talking about risks that are 5 to 10-fold higher for very common diseases like obesity, diabetes, heart disease, blood pressure, hypercholesterolemia, premature death from an exposure that was a low-calorie exposure in utero manifested 50, 60 years later after a lifetime of other exposures that contribute to that outcome and presumably are the result of epigenetic modification of metabolic pathways that are preparing that fetus for a condition of scarce resources after birth, but in fact that fetus was born into a condition of plenty, and the mismatch between plenty and metabolic sparse expectation resulted in this disease outcome later in life. So this is an epigenetic phenomena. It is actually very significant in terms of risk. How do we grapple with this complexity of a 6-decade time lag between the exposure, and that exposure is within the range of human experience broadly defined. There are lots of starving people in the world right now. So it's a normal exposure in some sense, but when extreme, clearly produces adverse effects. There's a huge time delay, and there are lots of other inputs into that outcome having to do with diet during the 60 years of life. This is very complicated, and it's very complicated for us to fit into our adversity paradigm ultimately. So what are some, I throw that out there, it's a bit of a sidestep, but it is something that's not going to go away that we need to understand. And we actually need to understand how chemicals might have similar influences, and they do, to calorie restriction. There are examples of that. And how do we incorporate our risk assessment in vitro of that kind of phenomenon? So here are some of the components of what we might use as information to determine adversity. If we focus on the toxicity pathways here, if we activate deleterious pathways, if the activation intensity takes off, becomes much more intense, if the spread in the activation by the compound exposure goes from one pathway to another pathway so that an entire network fires off, and if that activation is in fact irreversible, these are the kinds of components at a quantitative level, when we understand the system interactions, that will inform us about what adversity is in the cellular test system. But this is not going to be a simple problem. And I take us back to this figure because, just to reemphasize, in an animal model, we can induce a range of what is normal and a range of response by dietary manipulations. We can do the exact same thing in cells in culture. So the conditions that we choose for the culturing of our cells are going to be setting up the same kind of range of what is normal, what is acceptable, how do we deviate from that to what is actually adverse. It's going to be complicated to work out this issue. It's a very important issue. In fact, it's going to be what triggers the points of departure in our understanding of that. Here's another way to look at it. As you go from basal function in a pathway of response and then activate that pathway with exposures that can clearly be handled by the cell, as the activation intensity increases, as that spreads to other pathways, we move from what we will end up calling an adaptive response to an adverse response. And again, this is going to be highly contentious. Ultimately, this is where the rubber meets the road and where we end up identifying our points of departure. All right, so I'm on to my last two slides. These are summary points for us to think about. I really think it's important to not overpromise and fail to deliver. This is going to take us a couple decades. We're going to gain knowledge, over time, and we're going to feel better about our understanding of this. But it's going to take a while, and it's going to take meetings like this to keep the conversation going and a lot of work out in the field. And there are going to be lots of missteps. There needs to be some guidance, I think, some overall guidance from meetings like this or other bodies that get created to provide sharing of information and how the best approaches are developing. So the effort has to have some centralization to it. And it's not clear to me yet that we have any. But we need to be thinking about how that centralization is created and what it does and how it works. And we need to be focusing, at least at the beginning, on the commonality of responses and on pathways, and we need to be focusing on filling out this response landscape. That's going to take time to understand what the boundaries of that response landscape are. And ultimately, developing this systems approach is coincident with the progress in human health and medicine to a systems biology understanding of health and disease, and that will inform our toxicity testing as well. So taking from the lessons learned from the Human Genome Project, the goal is to build the best teams to do this work. It has to be driven by the science. So we have to keep ourselves informed of where the science is and how the science actually is working. But this is ultimately a very large management problem. We're talking about generating test systems and platforms that are coherent, that are validated, that work in different labs in different places, that give us information that's robust. So that is, to make that all happen, that's a very large management problem. It needs to be international because this system needs to be accepted universally, and there have to be milestones along the way. And these are all the points that the Human Genome Project learned as it was developing its approach to sequencing, which changed massively over the time of the 10 years that the Human Genome Project was happening. And finally, technology really matters in this. We don't know what the platforms are, what the visualization tools are, what the functional genomic integrating systems are. We don't know them because we don't have the data yet. That's going to be the input to those technical tools that we need to actually resolve this. So just a final couple of comments. I made this presentation to my lab group yesterday, and they asked two questions. They asked, well, how do you go from the past system, which we don't really believe in anymore, of animal-based toxicity testing and its limitations? How do you, what is that actual transformation? Is it gradual? Is it suddenly that doesn't work anymore and now we're doing something else? And actually that's a really good question. I don't know the answer, but we ought to think about it. What is, I assume we're going to bring in some of these tools that are going to inform the process, and ultimately the new tools and the new process is going to be the process. That's going to be part of what happens over the next 10 to 20 years. But it's actually a reasonable question. How do you do that movement from the process we're all still doing to the new process? And the second question is, how do you pay for it? Because this is going to cost a lot of money, and it's going to take a lot of time. Where's the money going to come from? How's the money going to be organized and directed to where it's needed to actually get the work done? And some of that's happening already, but I would say to a very large extent the academic world has not engaged in the process of generating the technology, tools, platforms, assays that are needed for this transformation to occur rapidly and robustly. So I think that's a really good question. Where's the money going to come from? Is it going to come from new TSCA regulation that includes a component that actually supports the development of this new knowledge? That would be nice to think of. But in this kind of funding environment, finding any new money to do anything could be really difficult. So I'll leave you with those two questions in mind. How do we make this change while we're learning the new system? How do we implement the actual risk assessment change process? And how do we pay over the next 20 years for this change to occur? Thank you very much. Hi, George. One at a time, please. My mind is sort of frazzled here, so. Okay.
We're very much looking under a lamppost, and so I'd welcome your thoughts on how we could broaden this out. I actually, my own idea is a very cheminformatic-driven development.
I missed that. What your own idea as well?
Is a cheminformatic-driven development of an ontology of presumed modes of action, and I think that there are reasonable ways to do that, but I like your thought on it as well. The second is this comment about complexity, and whereas I certainly agree with you that these pathways are complex, I think we really run the risk of getting to the point where we are characterizing complexity for complexity's sake. So I don't really need to describe every grain of sand to know what the soil is, or even surmise that I'm at the beach. So those are that I'd also like your thought on how much is enough. And then the third is really more of a challenge than a comment, which is I don't think that there is such a thing as a toxicity pathway, or if there is, it's very rare, and that virtually all of the interactions are interactions with pathways that are important for normal function, and therefore there aren't things that we can just distinguish easily. Therefore, the whole question of whether something is safe or not is going to have to be answered by models that I don't think we're thinking enough about, which are how do you translate quantitatively the effects at these molecular and cellular levels that we are getting increasingly capable of describing with what will likely be a very nonlinear effect that will lead to a biological response, either adversity, let's just leave that aside, but something that is really at the level of the organ or the organism. So and again, I have some ideas about that, but I'd be interested in what your ideas are on how to make that jump, because I think that it's the piece that we completely don't gloss over when we talk about new paradigms.
Well, those are really really good questions, and so the looking under the lamppost issue, I tried to approach that with the agnostic, discovery-oriented look for what the biology response landscape is, because I totally agree with you. I would argue that we can exercise the system and develop some of the tools by looking under the lamppost, but we can't know what the whole response pattern is going to be and what the risks are, basically, without having a broader understanding of what the overall biologic response landscape is. And that isn't just under the lamppost. And so that's why I was arguing for just basically doing stuff. And as you do stuff, you compile it, you put it into databases, and you bring it together in somehow, but you do it without prejudice, as opposed to using experts who have all their prejudice to design the system. So I actually agree with you, and I think it's important. The complexity issue is, it's a complicated question. So I tried to make the point that it's going to be overwhelming at first, but I think ultimately it's going to be quite simple. And I'd encourage all of you to look at the Ted Berlow TED Talk, because he actually deals with this question very well. So as all of that complexity comes in, what emerges from the understanding of that complexity as it's integrated in a systems biology way are the key nodes that actually inform the most about what the system response is. And finally, on your point of toxicity pathways, I totally agree with you. That I think I can claim ownership of having chosen that name and having labeled that toxicity pathway, taken that label and put it onto what we were trying to do in the National Academy of Sciences Committee. And it's received a lot of attack. On the other hand, it serves the same kind of function that cancer pathways have served in allowing us to focus in on prototypes as we develop the tools to thinking about how the system works. Ultimately, I totally agree with you. There is no such thing as a toxicity pathway per se. They're all biologic response pathways. And as we understand more of the landscape, the need for labeling something a toxicity pathway essentially goes away.
Bill Farland. Kim, great talk. I'm really glad you brought up the issue of the transition between current approaches and the new biology that we're looking at. One of the issues I think that we take on when we form a committee like this that talks about science to inform decision making is that there will be some way that we can make this transition at the same time make decisions. That's tough. I think that we've certainly been, as risk assessors, accused by our critics of paralysis by analysis. And so I guess I'd like your thoughts on what we tell decision makers as we begin to bring some of the new biology into the discussion of risk assessment.
Yeah, so I think it's a real problem because we've relied on a system that a large group of us now think is broken in a sense. It doesn't. That's why I began the talk, talking about how science is a cultural investment in what we think is true, and then that can change very suddenly, and it's not true anymore. And I think that's what's happened with our past system. And so, you talked about informed, so it needs to be an informed process of transition. One of the pieces of information is that, in fact, the old system, didn't work so well, and in fact, many of the components of the new system are equally predictive as the old system. And, we need to accept that, talk about it, and be okay with that, because it's true, and demonstrate it in various ways. And then I think what's going to have to happen is that we bring in the new tests, run them through a risk assessment process and evaluation process, and it gives similar results, similar safe, so that we're comfortable. And maybe there are some kinds of platforms that are actually intermediate between the past and the present that let us do that bridging.
Hi, I'm Pat Cassano, GE. I have two questions. The first relates to your distinction between screen, then test, or screening within the test system. Because as a practical matter, I want to do the screening first in order to determine whether I need to do the real test. So if you could explain in a little more detail maybe what the distinction is between the two. And then the second question relates to your point about focusing on cellular endpoints and tissues and organs being important but not necessary. I'm with industry, so of course I share the fear that if you focus on cellular perturbations, everything becomes hazardous. So how do you address that fear?
Well, first on the screening, so what I imagine as we learn more and develop this real test system that is systems biology based is that in fact some of the components of that real test system will be particularly informative. Some of the nodes of information that come out of that will be particularly informative, and that those will become the screening tests. And there can be components of that that are particularly informative. We're going to use QSAR, we're going to use read across, we're going to use all the information that's available to us, and that can be integrated into the systems biology approach. What I think is important is that it have the same conceptual and technical basis, that it is the same system in a sense, but you're hand-picking components that are easy, cheap, fast, and somehow universally informative for your screen. I forgot the second part of your... Oh.
The cellular endpoints.
Yes, the cellular endpoints. So you're asking the question really about how do we define adversity, I think. And so I told you, we don't know how to do that. And we don't know how to do it in the cell system yet. I believe we will figure it out as we get enough information. And I understand the fear, but it doesn't actually, it doesn't help to just then extrapolate from the cell system to what we aren't testing anymore, which is actually tissues and organs and organisms. It doesn't make that difficulty in identifying adversity go away. You still have to do it. And you still have to do it with the knowledge that you have. You're just putting, if you do that extrapolation, you're just putting more noise between your identification of adversity and the actual data.
Beautiful talk, Kim. Lauren Zeiss from California Environmental Protection Agency. I have a question about the safe, non-safe dichotomy being the holy grail. I thought your example for BPA and soy and casein was very interesting in that regard, especially when you think of things at the population level. Some people will be more like your rats on soy. Some people will be on bacon and eggs, maybe not casein, but bacon and eggs, and other things. So we have very varied exposures in the population. And if we think about it probabilistically, perhaps we don't need to move to a complete safe, non-safe threshold for looking at information like your comments on that.
Yeah, I totally agree with that. That's why I changed it to plausibly safe and plausibly not safe, because I think we have to incorporate into, it's no longer a bright line. We have to incorporate our uncertainty into our estimates of what the risk is. That's the bottom line. And I have no idea how to do that, but that's your job.
Thanks, Kim. This is provocative as usual. Stan Barone, US EPA. My point is probably more of a comment than a question, and it goes to you brought up the importance of biology as not being linear, and then you laid out several linear pathways, and I think in particular what struck me was adaptation linear to adversity. And I think the Academy, when I read the report, really talked about context, and I think at least my concern, not only as a cell biologist, neurobiologist, but also as a risk assessor, that reductionist approaches generally end up with a linear output also. So I think when we throw linearity at approaches of risk assessment, there's also linear approaches being employed in the reductionist approaches. So I guess I'm concerned, and I want to hear your thoughts on how we deal with context, life stage with respect to adaptation, because at least from my laboratory experience, adaptation isn't always a good thing.
Yeah, I mean, that's a great question, and of course there's no real way to answer that. But so what I was thinking about when Stan thought about that question was, there are people out there who are P53 heterozygous and xeroderma pigmentosa A deficient. And those individuals, that's part of our population response landscape. Those individuals, it doesn't matter, you look at them and they're going to have a response that's adverse. So there's a real difficulty in characterizing, and it's going to be probabilistic is what it comes down to, and we're going to have to figure it out. And we don't have enough information to know, and I would agree with you, it's not going to be linear. It's going to have to be very complicated in its interpretation.
Cheryl Walker, Institute of Biosciences and Technology. Ken, that was just a pleasure listening to that talk, and I want to congratulate the organizers for inviting you to kick it off. Obviously, fantastic insights and lots of thought that you've put into this. I have two questions. So I was intrigued by your early proposal that, and I agree, that we could really get bogged down by trying to now retranslate the systems biology in vitro approach back into animals, and I think that's probably right. So my first question is, how do you really think that it is unnecessary, or it will be circumstantially required? And I'm thinking, for example, of the old Ames tests, and where if it was a mutagen, it was carcinogen, and it really wasn't until some of these things were put in an in vivo setting that we realized, well, really there was a lot of exceptions to that rule. So that's my first question, and how are you thinking about that?
Oh, you're only going to ask one at a time, huh?
That's
really nice.
You're welcome.
So it's going to happen. So that extrapolation, there's going to be a whole group of people who that's all they do. To take the information, the systems biology, cellular level of information that's being generated in medicine and in this toxicity testing, developing this toxicity testing paradigm, taking that information and trying to understand how that then maps onto organ, tissue, organism responses. Mm-hmm. So that's going to happen no matter what. I guess my thought is, and my recommendation is that we not get bogged down in that. That's going to go on, and that's going to be helpful and informative. And it's going to be informative about the risk assessment process that takes place at the cellular integration level. But we don't have to do it.
So you see that occurring on the margins rather as being a central focus?
I wouldn't say it's on the margins. It may well be a very fundamental science to develop that integrated understanding of how cellular processes interact to give organ, tissue, organism phenotypic responses. But it's not necessary for us to do to understand the response to a chemical.
Okay. So then my second question, and I was actually very glad to see you put the Dutch hunger winter up there. As you know, that's a particular interest of mine. And, the sort of the 21st century interpretation of Paracelsus is the dose and the host make the poison. So in terms of thinking about these batteries that you might want to put together to capture the universe of cellular changes, and I'm probably inappropriately paraphrasing you, how do you see taking into account how those might change very dramatically over the life course? And does that now suddenly make this infinite, or is that just something that we don't really know how to deal with yet and we've got to figure that out next?
Yeah, I think that's very similar to Stan's question. And it has to do with host susceptibility and what the range of that host susceptibility is and how we incorporate that range into our assessment process. Well, first into our measurement process, but then into our assessment process. And I think we have to include it. We have to understand what that range is, and we're still grappling with understanding that. And Lauren's point was that, in fact, there are exposures, ambient exposures happening all the time to food across cultures that are radically different. And we know from our human disease phenotypes that, in fact, disease incidence is radically different in some cultures compared to others. We need to understand that better and incorporate that range of response over time. It's going to take time.
Thanks.